The discovery of penicillin, one of the most significant breakthroughs in medical history, began with a fortuitous accident in a London laboratory. In September 1928, Alexander Fleming, a Scottish bacteriologist, was conducting research at St Mary's Hospital. He had returned from a vacation and was tidying up his cluttered workspace when he noticed something unusual on a Petri dish containing Staphylococcus bacteria. One of the dishes had been inadvertently left open and had become contaminated with mold. Fleming observed that the bacteria surrounding the mold were being destroyed, while colonies farther away were unaffected. Intrigued by this observation, Fleming conducted further experiments to understand the mold's antibacterial properties. He identified the mold as belonging to the genus Penicillium and found that it produced a substance that killed a wide range of bacteria. He named this substance penicillin. Fleming realized the potential of penicillin as a powerful antibacterial agent, but faced significant challenges. The crude penicillin extract was unstable and difficult to purify, and his attempts to use it to treat infections in animals and humans yielded inconsistent results. Despite these obstacles, Fleming published his findings in 1929 in the British Journal of Experimental Pathology, highlighting penicillin's ability to kill bacteria. However, the medical and scientific communities largely overlooked his work, and it would take more than a decade for penicillin to gain recognition and be developed into a viable treatment. The next crucial phase in penicillin's journey came during World War II, when the need for effective antibacterial treatments became urgent. In the early 1940s, a team of scientists at the University of Oxford, led by Howard Florey and Ernst Boris Chain, took up the challenge of producing penicillin in sufficient quantities for clinical use. They developed methods to extract and purify penicillin, demonstrating its effectiveness in treating bacterial infections in mice and, later, in humans. With the support of the British and US governments, efforts to mass-produce penicillin were accelerated. Pharmaceutical companies, including Pfizer, played a pivotal role in scaling up production using deep tank fermentation techniques. By the mid-1940s, penicillin was being produced in large quantities, significantly reducing the number of deaths from bacterial infections among soldiers and civilians alike. The widespread availability of penicillin marked the beginning of the antibiotic era, transforming the treatment of bacterial infections and saving countless lives. For their pioneering work, Fleming, Florey, and Chain were awarded the Nobel Prize in Physiology or Medicine in 1945. The discovery of penicillin not only revolutionized medicine, but also paved the way for the development of other antibiotics, fundamentally altering the course of medical history. Please subscribe for more historical stories.